Dhruv. Thank you very much for inviting me. I'm very happy to talk to people and introduce the idea of cultivated meat, something that I believe very strongly in. So I lead a team of cell biologists that is trying to solve the problem of cultivated meat. And cultivated meat is a really difficult problem to solve because it has all of these different layers to it. So, for example, one of the layers is it's a complicated scientific challenge. This is the thing that drew me here in the first place, speaking as a biologist. But as anyone here who's been on a scale-up journey also knows, it's a very complicated engineering challenge because we have to figure out how to translate bench scale findings, often at the 5 milliliter to tens of milliliter range, all the way up to the hundreds of liters, potentially the thousands of liters. And that correspondence is really not clear. It's also a business challenge in some sense, because I think the cultivated meat sector needs to finalize on a business model that ensures profitability and sustained growth. And finally, from a consumer perspective, I think just the idea of cultivated meat really makes you really confront questions about what is the source of the food that we consume on a daily basis. So what I thought I'd do today is I'll tell you a little bit about what the process of cultivated meat looks like, sort of speaking generally, and then I'll zoom in on one or two tiny problems that, big problems, but I think we're getting close to the solutions that the cultivated meat industry is struggling with at the moment. So I moved to Europe about 10 years ago, and I have to say since moving here, I've only seen more and more of my friends and colleagues choose to be vegetarian or vegan or flexitarian. Despite this wealth of anecdotal evidence, if you look at the hard data, you will see that the per capita meat consumption in the EU has absolutely skyrocketed in the last 60 years. So each person consumes between 60 and 80 kilos of meat per year. Now, if you try to extrapolate this to the rest of the world, and you say, hey, what if every person decided it's my right to consume this much meat? Well, then our crumbling food system will just totally collapse. We know it can't sustain it. And a big part of the reason why it can't sustain it is because meat consumption comes at an extremely high environmental price that we often don't really talk about or think about. So here's some, I would say, frankly, very shocking statistics. Over about 80% of the agricultural land that is currently in use is either used directly for livestock farming or indirectly, which is to say by growing crops that ultimately supply the feedstock. Most of the antibiotics that are used globally are actually used in the cultivated meat, they're used in the livestock sector. So when people, say doctors are overprescribing antibiotics, perhaps they are, but my suspicion is that this is probably a bigger source of this problem. If you try to do the, admittedly very fraught exercise of trying to calculate greenhouse gas emissions, here too, you will see that no matter how you slice this pie, the consumption of meat has a significant impact on the environment. So all of these questions led us to the question, can we actually redesign our systems of meat production to be somehow more sustainable? The idea we had was the idea of cultivated meat. We're not the only players here, but in our process, what we do is we start with a small biopsy from a cow. This is either a muscle or a fat biopsy. Other companies that also use embryonic stem cells or induced pluripotent stem cells. So you need some kind of initial starting cell material. We typically start with a 1 gram biopsy sample. Somebody takes this biopsy sample, then they drive it back to the lab really quickly. We bring it inside the lab, we chop it up, we digest it with some enzymes, and then we sort the cells based on antibody staining and some tags that we put on the cells. We sort them into muscle and fat progenitor cells. Now, these two types of progenitor cells, we then grow up separately in our serum-free medium. I said we start with a 1 gram sample of biopsy. You may be asking, well, fine, 1 gram, that sounds like an interesting research experiment. What can you actually commercialize with that? Well, the thing is, because every time a cell doubles, well, it doubles. So you go from 2 to 4, 8, 16, 32, etc. You have this geometrical expansion. If you look at this cartoon down here, you see that a 1 gram sample, we could theoretically get something like 10,000 kilos of meat in 35 population doublings, which is a completely normal and expected number of cell doublings to get. Okay, that's quite cool. So we can actually generate a large mass of product, but where do we grow it? Because you think 10,000 kilos of meat is going to require a lot of space. The first time we did this, which was in 2013, the first burger that was ever made, cultivated meat burger, it was something like 250,000 euros to produce at the time. We used giant tea flasks. Anyone who's worked on tissue culture knows what a tea flask is. But so we use these very special cell factories or hyperflasks, and all of this is required to make one burger. Now, obviously, that's not what we want to do. We don't want to replace cows in the field with giant walls of plastic. So we need a more sustainable way moving forward. And so what we do is we grow the cells in big steel vessels called bioreactors, and I'm sure you're all very familiar with. Bioreactors, we can either grow the cells on microcarriers with tiny beads, we can grow them as aggregates, or we can grow them in suspension. And here I'm showing you a 3D reconstruction of a confocal image that I made when I first joined the company about 4 or 5 years ago now. And what you're looking at in purple is the nascent myotubes. So you're seeing tiny, like, let's say, pre-muscle fibers sort of forming, wrapping around these beads, and they're floating inside the bioreactor. Now, this process by which satellite cells or muscle progenitor cells fuse to form myotubes is called myogenesis. And this is roughly what the process looks like. But because it's a photonics conference, I thought it might be a cool idea to show you some really interesting movies. So this is a movie that I acquired on a quantitative phase imaging microscope. So this is a totally label-free microscopy technique that uses differences in phases to generate contrast. This is extremely sped up, but what you're looking at here is the formation of these fibers. And as the fibers thicken, then we can also see that there's more protein accumulation. So in addition to muscle, we can also do the same process with fat. What you see here is conventional fat in the middle, and what you see on the right-hand side is the cultivated fat that we produce in our facility, which sort of looks similar macroscopically as well as microscopically. And if we look at the fatty acid profile, we see it also matches conventional fat a lot more closely than, for example, coconut oil. And this, we think, has an impact on the taste profile, which we've also sort of validated through internal taste testings and taste testings with external agencies. Okay. So that's how we do cultivated meat. And in the last couple of minutes, I'll tell you about 2 of the big problems that the industry faces. One is the use of animal components, because the whole point of this is that we want to get animals out of the food chain. Seems pretty backwards to now reintroduce animal components. So one of the big issues with cell culture medium for a long time has been the use of fetal bovine serum. We've got here, I've linked 3 papers that our team has published here at Mosa Meat, where we figured out how to get rid of the serum from the medium. We've also now working on getting rid of even more animal components, because it's sensible for us, because it makes our process cheaper, it makes it more reliable, makes it more defined, as well as it's more friendly from a regulatory perspective. So we have lots of incentive to try to develop this further. And the second issue is the issue of costs. Now, I started by telling you the first burger we ever made was 250,000 euros, and that's true. And you may ask, well, why was it so damn expensive? There are a couple of reasons. One is, of course, the plasticware and so on, but also the raw materials that we used. So we currently have to use pharmaceutical grade ingredients. What I show you here is a screenshot from the local supermarket chain here in the Netherlands. This is 1 kilo of sugar for 93 cents, whereas if you go to the Sigma website where we buy, our chemical reagents, it's 128 euros. You can see there's orders of magnitude difference here in the prices. A lot of this is because this is the only grade that's available to us. Some of it is because at this stage in our development, we really want to use the highest purity ingredients so that our scientific findings are robust and reproducible. This means that in the long run, we're a little bit more efficient with our R&D. Our long-term vision is, of course, to replace as much of this as possible with more reasonably priced ingredients that are at the food or feed grade level, which is what we think can really sustain this process. Another thing that we do quite a lot of is to share as much knowledge as we can. This is because we're still a very young company. The cultivated meat sector is quite young, and I think it benefits everyone to share as much knowledge as possible. So we very frequently publish. This is also one of the reasons I really love working here. And with one slide where you can see some examples of other cultivated meat products that various companies are in the process of trying to make. Thank you.
Dhruv, the question everyone has in mind, because we have a lot of money in the photonics industry, we can buy the burger. Does it taste well?
Yes. So I've been on the tasting panel. I've tasted early versions of the product and more recent versions. The earliest version I haven't tasted because I wasn't there, but I was told it didn't taste very good because we hadn't cracked the fact that we need fat for the flavor. The versions that we have today, they taste identical to just regular meat. We've had chefs taste them, we've had professional tasters test them, and in each instance, people are able to very clearly identify that this is a cultivated meat burger because it tastes so much richer and so much more like regular meat than a conventional plant-based burger.
I offer myself as a volunteer to taste the burger next time that we see each other. But in addition, what can we do to help you? We have here the entire industry for companies that make sensors, camera detectors, equipment manufacturers like XM Tech in the room. How can we help you?
Well, I think one of the challenges we currently have is really online monitoring of how the cells are doing within our bioreactors. I think it's a very difficult problem to solve. We have things like impedance probes, for example, to just measure whether the cells are alive. You can correlate an impedance measurement with a viable cell density. But this sometimes works, other times doesn't work super well. We have optical probes for things like, DO, dissolved oxygen, pH, and those work quite well. But we still don't really have a good way of saying, are these cells healthy or not? By the time we start seeing drops in our doubling times, it's already too late for an intervention. So we need something that can almost predict when the cells are about to start crashing.
Dhruv, a lot of people have smiled, and if the meeting lasted for 1 hour, I would make a round of people who can help you for the online monitoring. But if we cannot, remember that everybody who is in the room and everybody who registered for this meeting is going to have the opportunity to be introduced by email to Dhruv Raina. Your inbox is going to be quite busy in the next hours.